I am very touched by the remarks of Paul Nurse. I wondered whether we were going to disagree or agree, but as soon as he commented on the dictionary definition of physiology as a subject that had been, as it were, somehow edged out by other subjects, I realized that we were on the same wavelength, because given the title of my lecture, you can see that I believe that if it was ever true that physiology had moved off center stage, it's coming back with a vengeance, and this Congress, in its many brilliant lecturers and symposia, will show you the extent to which that has happened and the extent to which it is riding on the back of some very fundamental reinterpretations of the major principles of biology. The lecture has already been published in the journal Experimental Physiology, and although I'm not adept at doing this kind of thing myself, I believe that if you point a camera at that picture, you can then get the app that gets you to the journal. Anyway, you can find it online very easily. So, first of all, a little bit of history in relation to evolution and biology. About 200 years ago, the great French biologist, he was actually one of the first to call biology biologie, to define the word, he published his book Zoologie philosophique, in which he established the transformation of species, which is, after all, a fundamental aspect of evolution. He's usually remembered for having at least repeated the idea that there was the inheritance of acquired characteristics, and for that reason that is sometimes called Lamarckism. I believe that is a wrong historical designation. He didn't invent it, he assumed it, as did Charles Darwin. Because when Charles Darwin published his Origin of Species about 50 years later, you will find about 12 references to and approval of the idea of the inheritance of acquired characteristics in his book. And in the preface to the 4th edition of the Origin of Species, he made it crystal clear that he thought that Lamarck was a great biologist. Darwin, of course, introduced the concept of natural selection, which has been the fundamental basis of evolutionary biology ever since. And then we move forward, another half century or so to Weismann, and the idea which he developed that in some way the germline was completely isolated from the organism and the environment, which of course would make Lamarckism, or the inheritance of acquired characteristics, completely impossible, and that was the fundamental assumption of what then became called neo-Darwinism, which started at that time. As you can see already from what I've said, Darwin was not himself a neo-Darwinist. And this was put together with, of course, much more recent studies of the nature of genetics, including a rediscovery of Mendel's experiments on genetics and Mendel's laws, put together into the modern synthesis in around the 1940s. Now, the main point of my lecture is to indicate that some of the ideas of evolutionary biology based on the modern synthesis and popularized by the selfish gene can be summarized roughly as follows. First, that it is a gene-centered view of evolution. It is the idea that there's a series of random changes in the genome which then accumulate slowly to allow selection to produce new species. It, of course, because of the Weismann barrier, and later on, of course, the central dogma of molecular biology, or rather I would say misinterpretations of the central dogma of molecular biology, it inhibited, or rather prohibited, or made it impossible for there to be inheritance of acquired characteristics. There was also, particularly in the book by Richard Dawkins, a distinction made between the replicator, the genes, later of course identified with DNA, and the vehicle which carries the replicator, the phenotype. And as I said already, the ideas were buttressed by what I regard as misinterpretations of the central dogma of molecular biology. And the whole point of my lecture, and what I'm going to tell you about now, is that all of these assumptions and rules have been broken. So, are mutations random, first of all? I would recommend people who still think that mutations are random, that it's chance that produces the variation in the genome, to read Shapiro's book, Evolution, A View from the 21st Century. It is absolutely full of the references showing that, as he says, it is difficult, if not impossible, to find a genome change operator that is truly random in its action within the DNA of the cell. All careful studies of mutagenesis show statistically significant non-random patterns of change. It's important here to ask the question what we mean by random, though, because it is not just a question whether the changes are truly random. It actually is very difficult to define what would be truly random. You have to ask random with respect to what, but rather whether the changes are functionally relevant. That's the key. And that's the reason why in the written version of this lecture, I have made it clear that I will use the definition that the changes are assumed by the modern synthesis to be random with respect to physiological function. And it is this that excludes the phenotype from in any way influencing or guiding genetic change. The experimental work that I will show you is that this assumption is now insecure, since some genome changes are related to physiological function. Let's take just one example of the many given in Shapiro's book. P-element homing in fruit flies, which was discovered around 1990. These are DNA transposons that insert into the genome, and some of them do so in a functionally significant way, in the sense that there is a greater insertion into regions of the genome that are related functionally. A possible explanation, though I don't think anybody has yet worked this out, is that the donor element and the target site may be brought close together in the nucleus by the nuclear proteins or interaction with the nuclear proteins, in which case the organization of the genome and its proteins is also important. This kind of information is therefore also inherited. And we can then move on to the question, were gradual mutations, gradual chance mutations, the way in which all proteins evolved? It's clear, at least in two cases, in two classes of proteins, that this can't be true. The 2001 Nature report of the draft human genome, comparing the human genome with other species, all the way from the fly through vertebrates, various kinds, through mammals to the human, showed, using the information on transcription factors and chromatin binding proteins, that the two classes of protein did not evolve one amino acid at a time, but rather there must have been shuffling of whole domains of proteins, one into another. This is from the Nature report of 2001, several proteins shown there for yeast, worm, fly, vertebrates, and human, and the stars show the various domains that have clearly moved around in the course of evolution as whole domains. That is not possible just by random accumulation. There has to be some mechanism of reorganization of the genome. So my first conclusion is that proteins did not evolve via gradual accumulation of mutations, and some other form of genome reorganization must have occurred, and there must therefore be something wrong or incomplete with neo-Darwinism. Now, I would argue, and I'm going to show that now, that the errors are both philosophical, that is, conceptual and linguistic, and they are scientific, that is, they have been shown to be errors by experiment, most of them more recent experiments. The selfish gene, was it actually a metaphor or empirical science? In response to a philosopher, Mary Midgley, who analyzed the selfish gene from a philosophical viewpoint, in 1979, Richard wrote a reply saying that was no metaphor. I believe it is the literal truth. And then he went on with a very interesting remark. Provided certain key words are defined in the particular ways favored by biologists. Now the question is, what is a metaphor? A metaphor does not cease to be a metaphor simply because one defines one key word to mean something different from its normal meaning. Indeed, it is the function of metaphor to do precisely that. I don't have time to go through the full detail, both of the philosophical analysis and of the scientific experimental analysis, but you can follow the arguments in that Journal of Physiology article published just 2 years ago. So, my conclusion is that the selfish gene idea, or the selfish gene way of representing neo-Darwinism, is simply not falsifiable. And the basic reason for that is that selfishness cannot be defined as an intrinsic property of nucleotide sequences independently of gene frequency. And it's a strange hypothesis that uses its own definition of its central entity as its one and only prediction. And it's important to note also that selection has actually never led to the formation of new species. Selection, as Shapiro puts it, operates as a purifying but not a creative force. After all, thousands of years of domestic selection have produced new varieties of dogs, cats, fish, and so on, but not new species. By contrast, hybridization, which is, as it were, mixing genomes up in various ways, does achieve speciation. So my next conclusion is that selfishness, however you want to interpret it, is simply not a testable hypothesis. Now the question is, why? Because you know, Richard Dawkins' books are extremely convincing. Anyone who's read his work will be amazed and has to have admiration for the way in which he convincingly writes what he writes. And I think that there is nevertheless an unraveling of the major thesis because the concept of a gene has changed. And this is a central part of the lecture. The concept of a gene has changed for the following reason. When it was introduced by Johansen in 1909, it referred to the inheritance of a discrete phenotype characteristic. Of course, the genome was not then known, DNA was not known. Notice that it was a definition based on the phenotype. The modern definition, of course, of a gene is now a molecular biological definition. It is a particular DNA sequence. And the important point to note is that these are not the same. The only way in which they could be the same would be if that vertical arrow on the right were the way in which DNA relates to phenotype. A gene for this phenotype, a gene for that phenotype, a gene for the next phenotype, and so on. We know, first of all, that there aren't enough genes for that to be true, and in any case, it isn't the way it works. As Paul emphasized in his introductory remarks, there is complexity in the networks, and of course, the networks are these parts of my diagram. These are the complex interactions that Paul Nurse was talking about. And so, it's incorrect to think there's a direct route from DNA to phenotype. What there is three-way interaction involving the various networks between DNA, the phenotype, and the environment. Moreover, if you do a knockout experiment, you will find in many cases that it doesn't reveal the function, because what is happening is that the network is buffering the knockout effect. There is a change, of course, the level of proteins, which proteins or RNAs are present or absent. The mechanism, though, is that if an organism cannot make product A by mechanism X, it does it by mechanism Y instead. You may therefore need to have 2 or even more knockouts to eventually get through to the phenotype. Incidentally, this view of biological network complexity has huge implications for the healthcare industry, which I don't think are fully realized yet, because the same principle applies to discovering new medications for complex diseases. You need multiple synergistic, that's the key, interactions, and the interventions therefore have to be synergistic and multiple, and that is why I believe that multi-action remedy is necessary. It's why the magic bullet approach simply doesn't work, and the results, disappointing results in general, of the pharmaceutical industries over the last 20 years show exactly that. Now, what's the evidence for what I've indicated? Let's look at some examples. This is an example from my own work looking at the pacemaker rhythm of the heart with a model that simulates the pacemaker rhythm. That's the electrical change at the top. We're showing two of the major proteins involved, the protein called IF, sometimes called the funny current, and a protein called the background sodium channel. And what we're doing here is knocking down the background sodium channel, which in this simulation initially contributes about 80% of the depolarizing current. And as you can see, as you knock it down, the change in frequency is actually very small. It's about 10%. If you used a knockout experiment to define the function of that particular protein and therefore the gene that is responsible for it, you would come to the false conclusion that the protein contributes only 10% to the function. It would not be true. And it's not true, of course, because the other mechanism, the IF mechanism, is kicking in. Moreover, the physiological analysis of that complex interaction leads us to understand why the buffering occurs. Because if you draw a line along the bottom of, initially at least, at the bottom of the electrical trace, you can see that what is happening is that the electrical potential is hyperpolarizing a little as a result of the knockout of the sodium channel, and what is happening then is that because hyperpolarization activates the IF current, it kicks in to produce a frequency that is very similar to the frequency at the beginning. So this is just a strange result that comes out of Dennis Noble's experiments and models on the heart. The answer is given by this lovely study by Hillenmeyer and his colleagues they were looking at the 6,000 genes in yeast, progressively knocking them out. 80% of the knockouts are silent under normal physiological conditions. You can only reveal that there is a phenotypic function by stressing the yeast metabolically. When you do that, it's like, in its sense, a metabolic stress is like a knockout. You're removing the possibility of getting a metabolic product by one route. The yeast has to try to do it by another route, and a knockout then may damage the physiological function. So this leads me to a very important question. Why should a physiologist even be concerned about evolutionary biology? Actually, if you look at the programs of international congresses of physiology over the last 30 years or so, you'll hardly ever find a symposium on biology of evolution. This is one of the first for many, many years to do that, and there's a symposium tomorrow in which some of the leading workers in this area will be presenting. The reason, of course, is that, as I emphasized at the beginning of the lecture, the modern synthesis is a gene-centered theory of evolution, yet organisms, as we've now seen, are very good at immunizing themselves from their genomes. And if you want to read about the consequences of that for physiology in general and for the nature of biology, then the article published there in the Royal Society journal Interface Focus is the place to look. The conclusion, therefore, is that knockouts do not necessarily, they sometimes reveal important results in relation to phenotypic change, but they rarely reveal regulators. And that article in Bioessays is an article worth following up on that. What's the nature of the problem? The nature of the problem, as Paul Nurse emphasized in his introductory remarks, is that there's biological complexity in the networks. We have forms of downward causation. Of course, there is also upward causation, the traditional reductionist view that genes code for proteins. That's why I've got a different type of arrow at the bottom. It's a coding step, and then they form pathways combined with lipid elements to form subcellular and cellular structures all the way through tissues, organs, and organism, and so on. And it was this view, of course, that led Richard Dawkins to his statement in the Selfish Gene, they, that is genes, created us body and mind. It just simply isn't true. I prefer the statement from the Nobel Prize winner Sidney Brenner, who at a major meeting of the Novartis Foundation in 2001 wrote, "I know one approach that will fail, which is to start with genes, make proteins from them, and build things bottom up." What he had in mind was exactly what I'm going to show you now, but it's very familiar, I know, to physiologists, which is that in addition to the standard reductionist view, you've got to add all the forms of downward causation, including systems-level triggers of cell signaling, system-level control of gene expression, and you'll notice that I reverse the bottom arrow. The truth of the matter is that it's protein machinery that reads and corrects genes. And of course, as we now know, there is epigenetic marking at all levels. Excuse me. If you want to follow up many of the aspects of downward causation that I can't deal with in a lecture like this, then this focused issue of the new Royal Society journal, Interface Focus, has a whole series of articles on this area. So, if I express this in terms of what I've sometimes called the principles of systems biology, one of them is to say that the transmission of information is not one way, and therefore I would say that the central dogma of molecular biology has been misapplied, interpreted, misinterpreted, or even incorrect. It depends on what you define it as being. In the form in which Crick originally showed it, of course, which is that DNA forms a template for the formation of protein, but protein sequences of amino acids do not form a template for the formation of DNA, then of course it's absolutely correct. It is the interpretation that transmission of information does not go the other way that is incorrect. Now it's very interesting, some of you will already have realized that if there is downward causation, and particularly downward causation onto the genome, then the spectre, if you want to call it that from the point of view of a modern synthesis, or the interesting phenomenon from the point of view of a physiologist of Lamarckism comes back into play. And it's very interesting that one of the best thinkers who contributed so much to the development of the modern synthesis, that is John Maynard Smith, wrote in his book in 1998 that Lamarckism is not so obviously false as is sometimes made out. Those who want to follow up the history of that particular remark and what it led to can find that in the published lecture in Experimental Physiology. Moreover, DNA can't be the sole transmitter of inheritance. After all, we all inherit a complete egg cell together with all the marking and systems that are inherited through the egg cell. I think we should reverse the usual question. It's what normally prevents the inheritance of acquired characteristics rather than the other way round. Examples of experimental evidence for inheritance of epigenetic information is just now becoming a flood. Here are just some of the examples. Weaver's work, I'll refer to in just a moment. The work of Skinner and his colleagues looking at endocrine disruptors inducing epigenetic transgenerational change. The work in this team looking at male line transgenerational responses in humans. Beautiful work which will be discussed in tomorrow's symposium by Yunhua Sun and his colleagues at the Fish Institute in China on cross-species cloning, and the recent work by Rehavi and his colleagues on inheritance in C. elegans. Let's have a look at some of these. The Weaver work was even featured on Valentine's Day in The Guardian a few years ago with the lovely headline, "Motherly love may alter genes for the better." What they showed was very interesting. It's well known that the stroking behavior and the licking behavior that rodents show to their young, and many other animals, of course, too, is a major factor determining the health state of the young as they grow up into adulthood. And colonies that do that a lot produce healthy offspring. Colonies that can't do it because, for example, of competition and predators produce much less healthy offspring. That's been known for a long time. The contribution of Weaver and his colleagues to show the mechanism is, on the one hand, a kind of cultural phenomenon, and at the same time a transgenetic phenomenon, that is to say, an epigenetic marking that can be transmitted down the generations. What happens is that the stroking behavior marks appropriate genes in the hippocampus that predisposes to that kind of emotional behavior. And as you will realize, even if that is not transmitted down the germline, it'll still be inherited. And the could be should now be replaced by are, because later work has identified where the marking is occurring. These are two more examples of epigenetic inheritance. I sense perhaps that time is pressing, so I won't go through those in detail. My conclusion at this stage is that with many examples that are now accumulating very rapidly, it's clear that environmentally induced changes can be inherited. And so I think it's important to ask the question, which is the question that Sun and his colleagues at the Fish Institute in China asked, what happens in cross-species clones? It's very clear what should happen. If you transfer the nucleus from one species into the fertilized but enucleated egg cell of another, you should find that the organism is defined entirely by the nucleus and its genome. So what they did was to take the nucleus from a carp, which is this central animal, and transfer it into the fertilized but enucleated egg cell of the goldfish. And the animal that was produced, is actually very rare that you get an adult from such transspecies cloning. What usually happens is that the embryo freezes at some early stage and then subsequently dies. You'll notice that in both shape and indeed in skeletal structure is intermediate, and that can be demonstrated by counting the number of vertebrae. In a goldfish, it would be about 26. In a carp, it's about 33. And if you look at the carp nucleus in the goldfish egg, the result is closer to the goldfish, about 28. And as they say in their paper, it's clear that egg cytoplasm plays an important role in regulating the somite development and vertebral number in the nuclear transplants. And actually, it's not a completely new observation. As a transspecies clone, it's certainly new and a fundamental breakthrough. But way back in 1957, Mickey and McLaren showed that if you transfer the embryo from a mouse strain that has 6 vertebrae in the tail into a mouse strain that has 4, you get an adult growing up with 6. I'm sorry, I got that the wrong way around. You get an adult growing up with 4. In other words, there is a maternal effect. There are whole books now on inheritance of maternal effects. Barbara McClintock. She got the Nobel Prize, I think at the age of 81, in 1983, for the discovery of transposons, that is, jumping genes, to use a more popular terminology. And in her Nobel Prize lecture, she has this, is published in Science, she has this lovely text. Future attention will undoubtedly be centered on the genome, and with greater appreciation of its significance as a highly sensitive organ of the cell. That needs underlining, I forgot to do it. Monitoring genomic activities and correcting common errors, sensing the unusual and unexpected events, and responding to them, often by restructuring the genome. She wrote at that time, we know nothing about how the cell senses the danger and initiates responses to it. Well, the evidence is that we do now know. as Burton and his colleagues in a recent book say, a genome consists largely of semi-stable genetic elements that may be rearranged or move around in the genome, thus in changing the information content of the DNA. So the next conclusion is that cytoplasmic changes can be inherited. What are the possible mechanisms? I'll just finish with two examples and then I'll sum up the conclusions of the lecture. This is from the work that I highlighted earlier on by Rehavi and his colleagues. They took the worm C. elegans, which can be infected by a particular virus. Not many viruses can do that. The worm, if it has the right DNA, responds by making a small RNA that silences the virus. So there's a silencing response which is acquired as a consequence of the infection. That, of course, has been well known for a long time. What Rehavi and his colleagues did was brilliant. They proceeded to cross the worms with that acquired characteristic with worms that were wild type, many of which do not have the DNA for making that particular RNA. And then, of course, the crossed ones in the first generation will have the ability. But what then happens, you go down the generations, if you do enough breeding, you will get worms that do not have the DNA, but who are descendants of the worms that had acquired the characteristic. And what you find is that the characteristic is acquired even though the DNA is not transmitted. And the mechanism is clear. What is happening is that RNAs are passing down through the germline. Those are then amplified up by RNA polymerase, and the inheritance is robust for 100 generations, a whole year, which is as long as they followed it. This work, even more startling. This is from the work of Joe Nadeau, who was at the time, I think, one of the key workers in the Institute of Systems Biology in Seattle. He's since moved away from Seattle. This paper, just published a few months ago in the Proceedings of the National Academy of Sciences, shows epigenetic effects dependent upon ApoBec1 deficiency that persist for many generations and are as strong, as they write it, as conventional genetic inheritance. The idea, which is the idea that some of the defenders of the modern synthesis have favored, that somehow all epigenetic effects die out quickly and therefore they don't persist and could not be the target of evolutionary change, that assumption has clearly now been shown to be incorrect, at least in some recent examples of that kind of inheritance. The PNAS even published a commentary article saying the belief that the soma and the germline do not communicate is patently incorrect. So my sixth conclusion is that environmentally induced changes can be inherited, the inheritance can be robust, it can carry through many generations, and in some cases we now even know the molecular mechanisms. So the question is, if gradual mutations is not the answer, what is responsible for the reorganization of genomes? The answer seems to be, looking through the experimental and evolutionary biology literature, there are many mechanisms. And in fact, of course, the people working with unicellular organisms have for many years known that genome bits and pieces can be transmitted across from one species to another, so much so that at the level for example, of bacteria, people don't any longer talk of particular species. The whole thing is a mishmash of interconnected network rather than the simple tree of life that was the basis of the modern synthesis. I had the great pleasure of interacting before her death with Lynn Margulis, who was one of the great innovators in relation to the idea that what we identify as physiologists and biochemists as the mitochondria in animal cells and chloroplasts in plant cells were in origin invading or absorbed or ingested bacteria. And I had the great pleasure of chairing a debate between Lynn Margulis and Richard Dawkins in Oxford University in 2009. That debate, incidentally, is fully recorded. It's on the website Voices from Oxford. So if you just type Voices from Oxford in Google, you'll get it, and you'll find the whole of that debate, it went on for about 4 hours, is there. And what I learned from Lynn Margulis and from many of the other evolutionary biologists that she introduced me to is that a sea change is occurring in the way in which we have to view particularly the early stages of life development on Earth, which has to be seen as a network rather than a tree. But the crossings even occur at the higher levels, as Shapiro's book shows. I referred to this book a little bit earlier from Burton and his colleagues. This is the quote: "The genes begin to look like hardly definable temporary products of a cell's physiology." So my final conclusion is that if functional changes in the adult can be inherited and therefore a target for natural selection, then physiology, that is the analysis of function, is relevant to evolution. Of course, it's always been so, because if you ask the reason why natural selection works in the way it does, it is that certain organisms function better than others. That aspect, the sort of post-genomic variation, has always required an analysis of physiology. What we're beginning to see now is that physiological function can have influences even on the origin of genetic variation. And that's the reason why in the article I write that it is hard to think of a more fundamental change for physiology and for the conceptual foundations of biology in general. So let's summarize up with a work towards a new synthesis. Instead of the gene-centered view of natural selection, of course genes are important. Don't misunderstand what I'm saying. Absolutely so. Part of the functioning, as Paul Nurse pointed out in his introductory remarks, in any network of biological interactions. But what we have to recognize is that selection is clearly multi-level. The impossibility of inherited acquired characteristics, that's clearly not true. In some cases at least, and the only question is how many cases will there be, some acquired characteristics can be inherited. Distinction between replicator genes and vehicle. I've never managed to understand this distinction. I know what Richard Dawkins meant, but somehow it's putting the cart before the horse. The reason, the logic, to use the logic of life way of looking at physiology, why one organism survives and another doesn't, is functional. It's functional at the level of the phenotype. Somehow or other, to regard the phenotype as just a vehicle for its genes is, it is philosophically difficult to accept. And of course, with Barbara McClintock's way of putting it, we should see that the genome is an organ of the cell, not its dictator. Control, in other words, in those networks is distributed. Of course, the genome is very important, and very important also as a controller, but so are many other aspects of biological networks. And the central dogma of molecular biology, well, whatever one says about that and however it has been misinterpreted, I think we conclude that genomes are not isolated from organism and environment. And so in the article, I've suggested that we should replace the modern synthesis by a move towards an integrative synthesis with, as I've said already, selection being multi-level, with acquired characteristics being inheritable, a point that Darwin himself also admitted and used, that the genome is an organ of the cell and that genomes are not isolated from the organism and environment. So just a few references for those who want to read more on all of this. The article itself can be downloaded straight from Experimental Physiology. It's generated so many questions from people emailing me and then also taking part in blogs on the internet that I've just written a set of answers to the most popular questions. So if you've got, as it were, simple questions about what I've been lecturing on, you can find the answers to many of them on that website. And then further reading, I would suggest a lovely book by one of the presenters at the meeting tomorrow, Gerd Müller and his colleague, on evolution, the extended synthesis. Gysis and Jablonka in relation to modern versions of what is sometimes called Lamarckism, and they have this beautiful title, Transformations of Lamarckism. Shapiro I've already referred to, a view from the 21st century. Burton and his colleagues I've also referred to already, the concept of a gene in development and evolution, and my own little book, The Music of Life. So with that, I come to the end of my lecture. Thank you very much.
So Dennis, thank you very much for a beautiful and thought-provoking lecture delivered as a true physiologist, that is, someone who is studying the philosophy of life. So thank you very much. And in commemoration of this beautiful lecture, I'd like to present on behalf of the International Union of Physiological Sciences this commemorative. So Dennis.
Thank you very much. Yes.